What came first, DNA or RNA? RNA is widely believed to have come first in evolutionary history based on the RNA World Hypothesis. This hypothesis suggests that early life forms used RNA both as genetic material and as a catalyst for biochemical reactions, known as ribozymes. RNA is simpler in structure compared to DNA, and it can both store genetic information and perform enzymatic functions. DNA likely evolved later as a more stable molecule for long-term genetic storage. Why is DNA the primary genetic material in most organisms, not RNA? Well, let's dive into a few reasons. Stability. DNA is more chemically stable than RNA due to the absence of the hydroxyl group at the 2' carbon in its sugar, deoxyribose, compared to ribose in RNA. This makes DNA less prone to hydrolysis. Additionally, DNA's double-stranded helical structure provides extra stability and protects its genetic information from damage. Fidelity in replication. The double-stranded nature of DNA allows for proofreading and repair mechanisms during replication, reducing the chance of mutations. On the other hand, RNA is single-stranded and lacks these repair mechanisms, making it more error-prone. Longevity DNA is better suited for long-term storage of genetic information because it is less reactive and degrades more slowly compared to RNA. Specialization RNA is more specialized for short-term roles, such as acting as messenger RNA, transfer RNA, and ribosomal RNA. DNA's role is primarily long-term genetic storage. How do cells differentiate between DNA and RNA? Cells distinguish DNA from RNA using several structural and chemical differences. Sugar molecule. DNA contains deoxyribose, lacking a hydroxyl group at the 2' carbon, while RNA contains ribose. Enzymes in the cell recognize and bind specifically to these sugars. Bases. DNA uses thymine, whereas RNA uses uracil. This distinct base allows enzymes like DNA polymerase and RNA polymerase to identify the type of nucleic acid. Strandedness. DNA is double-stranded in most organisms, whereas RNA is single-stranded. This structural difference helps cellular machinery to differentiate them. Specialized enzymes DNA and RNA are processed and handled by different enzymes. For instance, DNA polymerase exclusively interacts with DNA, while RNA polymerase interacts with RNA. Why don't living organisms have RNA as their genetic material? Well, RNA is more prone to chemical degradation and hydrolysis due to its 2' hydroxyl group. This instability makes RNA unsuitable for storing genetic information over long periods, especially in complex organisms. RNA's lack of a double-stranded structure and proofreading mechanisms leads to higher mutation rates. This is advantageous for some viruses but detrimental for stable inheritance in larger organisms. During evolution, organisms that transitioned to DNA as their primary genetic material gained a selective advantage due to its stability and error-correcting capabilities. As life evolved, RNA's role became more specialized, for example, in protein synthesis and regulation, while DNA took on the role of stable genetic storage. Why do some viruses use RNA as genetic material? RNA's high mutation rate allows RNA viruses to evolve quickly, helping them adapt to new environments or hosts. RNA viruses often have smaller genomes and simpler replication machinery, which suits their parasitic lifestyle. Additionally, RNA molecules can adopt complex structures and perform catalytic functions, giving RNA viruses certain functional advantages. In conclusion, RNA likely preceded DNA in the evolutionary timeline, but DNA became the predominant genetic material due to its greater stability, fidelity, and suitability for complex life forms. Cells differentiate between DNA and RNA through structural and enzymatic specificity, ensuring efficient functioning and specialization. RNA, while unsuitable for long-term genetic storage in complex organisms, remains vital in certain biological processes and as the genetic material of some viruses. Nature is ultimate,